So bifunctional degraders, because they're very large molecules, they are what we would consider beyond rule of 5 molecules. So they fall in a very different space to the typical small molecule orally drugs, as shown here in this plot. That plus this unique mechanism of action of an event-driven instead of occupancy-driven makes it very challenging to design this molecule. I want to propose the Schrödinger structure-based TPD design workflow, where if you don't have the ternary complex of your system, you will start by running product panel 1 to generate ternary complexes with 2 different methods. After that, you will be able to get within the top 20 reasonable poses, but those will not be, you will not be able to distinguish between the top 3 poses. For that, you will need the more accurate scoring MD methods, and we are going to release product panel 2 this year that will allow you to automate the scoring using metadynamics and MD Piper MMGBSA. And ideally, if you do have the ternary complex crystal structure and you are within certain constraints, which must be first, the KDs correlate with the DC50s, and the second one is that you're working with degraders that have the same exit vector than your ternary complex and similar linker length, since we cannot sample changes in the ternary complex during the FEP simulation. Then you can use FEP plus for linker optimization, and that's out of the box using the FEP panel. So the exit vector of a degrader is the direction in which the degrader exits the binding pocket towards the solvent. To sample this, we use Desmond. So in this example, I'm showing a BRD4, and we will be sampling only a truncated degrader without the second warhead. We are going to calculate the vector, so the exit vector for this ensemble, using 2 points. These are defined by the user. Typically, it will be the anchor atom and the linker terminal atom. And after clustering, we will be able to get the different exit vector preference for this particular linker, and we can visualize these exit vectors in Maestro. Okay, so I will be talking about 2 workflows, degrader sampling and protein-protein docking, followed by linker sampling. In this workflow, what we are doing to generate the ensemble of ternary complexes, we sample the isolated degrader with macromolar ComGen. Then we superimpose each proteins with respect to their warheads. By doing that, a lot of this initial ensemble will be filtered out, a lot of these conformations, if they're clashing with the protein, right? Protein backbone and protein side chains, and you can tune, if you want to allow some clashes so that you are not very rigid, in that initial filtering. So that will basically generate the ternary complexes, and then what we do at the end is we score the complexes in place using the Piper's scoring function. What this workflow allows you to do also, apart from, once you have that ensemble of ternary complex, if you have a congeneric set of degraders that share the same or similar warheads but different linkers, you might want to be able to compare the ensemble of ternary complexes between these congeneric degraders. And we do that by generating an ensemble of custom pharmacophores that depend on the relative orientation of these 2 warheads in the ensemble. For workflow number 2, which we call protein-protein docking followed by linker sampling. So this method is very different, but the outcome would be the same to generate ternary complexes. Here we start by running Piper with distance constraints to obtain possible PPIs between the E3 ligase bound to the warhead and the target bound to the warhead. After we obtain this very large ensemble, what we do is we sample the linker either with ComGen or Prime, and many PPIs will be filtered out in this step because they are incompatible with that particular linker. After this step, we will cluster the ensemble, and we can either do it with interaction fingerprints or RMSD, and then we will select what we call the winner cluster, which based on certain rules that we found empirically. And then we will refine and score ternary complexes using Prime. So moving to scoring method 1, the metadynamics post-scoring, we are using well-tempered metadynamics. The collective variable here is the distance between the center of mass of selected key residues on the target and the ligase. And what we use to score these different complexes is a normalized post-score. The second scoring method is Piper docking and MMGBSA score on top of MD trajectory. So what this means is that we run unbiased MD, so multiple replicas, and then we calculate either protein-protein MMGBSA score or the protein-protein Piper energy in place for each snapshot in the trajectory, and then we take the ensemble average. This panel is called the macromolecular pose filtering. It's in beta in 25-1, and it allows you to filter poses based on 3 different things, either properties of each entry. Here on the top right, you could also use residue property levels. And what I will focus today is using AGXMS data. Here on the left, what I'm showing is the typical experimental setup for getting experimental ternary binding affinity measurements. This is SPR, where one of the proteins, here in this case is the ligase, is immobilized. And then the binary complex, in this case, the target with the PROTAC, it's basically thrown through the chamber. And what you are measuring, this formation of ternary complex, it's an apparent ternary KD. And this is the result. So this is on the left, the crystal-like PPI, and on the right is the decoy PPI. So you can see that FEP plus can recapitulate the experimental ternary KDs when a crystal-like model complex is used, similarly to when you have the crystal structure and not with a decoy complex.
We have some models calculating the linker properties, the thorough sampling model, and then we have a substructure distance model as well, which calculates the minimum distance between two substructure groups, in this case, the warheads, and those are supplied as SMARTs or SMILES patterns. Very simply and easily do that within Live Design by clicking on this View 3D molecule here, and the 3D viewer will be loading. Then I can select the degraders that I want to display and then turn this on, and we can see that we can look at our degrader and see how it fits within the ternary complex. I can also overlay multiple degraders and see how they fit, how they interact with the protein, and also add my crystal structures as a reference as well.